Hi, this is Dr. Mark Poole with another Chalk Talk. I'm going to cover basic cellular reactions to injury in the CNS. As with most other organs, the CNS has a stereotypic pattern of cellular responses to injury. Because it's the CNS, there's a particular vocabulary that you'll have to learn. But the underlying mechanisms are similar to other organs. This is kind of my reaction to neuropathology usually. It does have its own particular set of vocabulary, and you have to learn some of these terms, but I'll try to make it as easy as possible so that we can learn this together and have it organized in our brains. It's useful to start off with the basic principal cell types of the nervous system, and then I'll go into the specific cellular reactions for each of these. Basically, you can divide the CNS into neurons and everything else. And the everything else, which is in the red font, are called glial cells. And this is a population of different cells. They have different functions and come from are derived from different stem cells. In the peripheral nervous system, it's much easier. There's just neurons and Schwann cells, which are the cells that produce myelin around the axons. This is just a quick review of the neuron. The cell body is what contains the nucleus. The dendrites are short processes that act as antennae to receive signals. And then the axons carry the action potentials, which we'll learn about in other sections of the course, which are very long and can go many centimeters to reach their terminal destination. And it's the axons that are sort of like the high voltage wires carrying information away from the cell body. Nissl substance are basophilic granules that are abundant in the cell bodies of large neurons and basically consists of rough ER. This is the, as you know, protein manufacturing process. These the Nissl substance can be lost in response to injury, and we call this centrochromatolysis. So this basophilia in the cell's cytoplasm will be lost. And this is the first term to explain what I meant by the CNS kind of having its own terminology. So the panel on the left shows an example of central chromatolysis, and notice how the cytoplasm has a homogeneous pink appearance, and the cell has lost that cytoplasmic basophilia because the Nissl substance is lost. Another characteristic injury is called eosinophil change or red neuron. This is an early change following hypoxic or ischemic injury, and I'll talk about that in much more detail when we discuss a case later on in the course where we're going over the vascular damage to the brain. Another set of neuronal responses to injury involves the acquisition of intracellular inclusion bodies. And these are useful to recognize as we go through the course because they often reflect or are more specific for the underlying disease process. And so as we go through this course, whenever there is pathology that has these characteristic intracellular inclusion bodies, I'll be sure to point them out because that will be a visual clue as to what type of what the disease process is. And these can be either intranuclear or they can be intracytoplasmic. In this case, there's an intracytoplasmic inclusion body. To demonstrate axons, we use different special stains. In particular, we use silver impregnation stains or immunohistochemical stains to demonstrate axons. Otherwise, they're they're not really readily visible on routine H&E stained sections. This happens to be a silver stain, and the silver precipitate that's in the stain binds to the axons and gives them this definition. Another stain that we use to indirectly look at axons is a Myelin stain. Now this stain usually stains the myelin blue. So in the upper part of this image is cerebral cortex, showing which is predominantly cells, and so this stains pink with an H&E stained section, and has little myelin because this is mostly the cell bodies and dendritic processes. At the lower part of the image, which stains intensely blue, is the subcortical white matter. And this stains intensely blue because this is where the bulk of the axons travel to reach their destinations. So grossly, if you think about it, the cerebral cortex is the gray matter, and the subcortical tissue is the white matter. Now the reason why these stains are useful is to identify syndromes that have in common demyelination. And this is processes that involve the destruction of myelin with relative preservation of the axons. So by using these different stains, on the left we can use a Myelin stain to note these pale zones that are interdigitated with the more darkly staining regions that indicate demyelination. And we can use a silver stain in the same way to delineate these pale areas in between where the streaks of axons are going through. And so we can identify both axonal loss with the silver stain and loss of myelin with the Myelin stain. Again, remember that this is not a specific disease, but a pattern of injury that we'll be able to see in different diseases as we go through. Another particular form of axonal degeneration is called Wallerian Degeneration. This is most often seen in the spinal cord due to some transection of the cord. Wallerian degeneration describes when an axon is transected, the distal stump becomes separated from the cell body and degenerates. So portions of the axon distal to the site of transection are disconnected and undergo degeneration. As the axons degenerate, their myelin sheaths undergo secondary destruction, and this is called secondary demyelination. And you can see this when you look at sections of spinal cord, such as in this set of images. Myelin, remember, stains intensely blue, so the image on the right shows a normal cross-section of spinal cord, and the image on the left shows pale areas indicative of secondary demyelination. So which section is below the site of transection and which is above? Okay, if you answered the section on the left is below the site of transection, that would be correct. Because remember, Wallerian degeneration involves loss of the axon and then secondarily loss of myelin that's distal to the site of transection. Very good. Okay, the next cell that I'll talk about are astrocytes. These are supporting cells that regulate the extracellular environment of the nervous system. They have some different morphologies, and I'll show you examples of these. These are fibrillary astrocytes and gametocytic astrocytes. So these function as a reaction to injury to protect the neurons. They also induce the blood-brain barrier, and they regulate the extracellular electrolyte environment and neurotransmitter concentrations. So they have very important regulatory functions. These are the normal fibrillary astrocytes, and it should be obvious from this special stain that's used to stain astrocytes why they're called fibrillary. The cell processes extend out, making connections to neurons, blood vessels, and other cells to coordinate the microenvironment and react to injury. Through the foot processes that surround capillaries or extend to the subpial or subependymal zones, they contribute to the burial functions that control the flow of macromolecules between the blood, the CSF, and the brain. So they induce the so-called blood-brain barrier. In reaction to injury, the nuclei of astrocytes become enlarged, vesicular, and develop prominent nucleoli. The previous scant cytoplasm, which is hard to see on H&E sections, becomes bright pink, as indicated by the arrows here. And the nucleus becomes sort of displaced to the periphery of the cell. And these are called gemistocytic astrocytes and are an indication of injury to the brain. Again, this is not specific to a particular disease, but is a pattern that's common to a lot of different brain injury. Another more general form of cellular reaction to injury that involves astrocytes is called astrocytosis. And this is where there's just an increased number due to proliferation of astrocytes. And this involves fibrillary astrocytes. We use special stains to identify the astrocytes. This is an immunohistochemical stain for glial fibrillary acidic protein, which is found in the cell body and processes of astrocytes, and is a handy stain for identifying an increased number of astrocytes in injury. Another term that is sort of used interchangeably is called gliosis, even though this is a little bit more general term, because usually with gliosis there's an increased proliferation of all glial cells, including astrocytes. And again, we can use the GFAP stain to identify increased numbers of astrocytes. In the brain, gliosis is just a term for scarring. And so, again, this is not specific to any particular disease, but is a common injury pattern. Next, we turn to oligodendrocytes. These are cells that form myelin sheaths in the CNS. These can generally be identified on H&E stained sections by their round nuclei with these clear halos that are identified by the arrows. The astrocyte nuclei in the background are generally less dark, and so that's how experienced neuropathologists, not myself, can readily identify these on H&E stained sections. The injury or apoptosis to oligodendroglial cells is a feature that is seen in acquired demyelinating disorders and leukodystrophies, which I'll talk about later on in the course. The final glial cells that I'll talk about are microglial cells. These are phagocytic macrophage-like cells that reside in the CNS. They are bone marrow derived cells from the monocyte macrophage lineage. It's difficult to see these cells with routine H&E stained sections, and we really only notice them in situations where there is cellular injury and they become activated. They can transform into microglial nodules, which is basically just describing an accumulation of these cells, or transform into histiocytic cells, which in the CNS, we have to have a special name for these, and so they're called Gitter cells. So I'll show you a couple examples of these. So this is an accumulation of these cells. You can note, just like in other organs of the body, These macrophages have irregular kind of indented nuclei, and when they form a cluster like this in the CNS, we call them microglial nodules. And these are characteristically found in chronic encephalitis. Again, they're not specific to any disease, but are a reaction pattern to injury. And these are the so-called Gitter cells, which are macrophages or histiocytes. And these are derived from microglia and circulating monocytes from the bone marrow, and are seen in necrotic or inflammatory lesions. And they have the same function as in other parts of the body to phagocytose cellular debris and dead cells. So that ends this part of the chalk talk. Again, this was just a general overview of cellular reactions to injury, and keep these terms and the images in mind as we go through talking about more specific diseases in the rest of the course.